Welcome back, everyone, to the Deep Dive. Are you ready to explore something truly foundational today?
I hope so, because we're going deep into the world of microbial eukaryotes,
Yeah, protists, algae, fungi.
the often overlooked ancestors and powerhouses of so much of the life we see around us every day.
I know, right? We're talking about organisms that have been shaping the planet for billions of years,
Mm.
much longer than macroscopic life has been around.
Exactly. Their story is one of incredible diversity, adaptation, and evolutionary innovation.
So for this deep dive, we're breaking things down into four key areas.
Okay.
First, we're going to look at the defining features of eukaryotic cells, their organelles, and the evolutionary relationships. Then we'll jump into the incredible world of protists, those eukaryotes that don't fit neatly into any of the other kingdoms.
Right.
Then we'll uncover the secrets of the fungi.
Essential, yet often underappreciated.
And finally, we'll explore the Archaeplastida, the lineage that gave rise to all plant life.
Sounds like a plan.
And our mission here, as always, is to uncover the most important nuggets about the diversity and impact of these fascinating microorganisms.
I'm ready when you are.
Okay, so let's start with the basics. What makes a eukaryote a eukaryote? What are the key features that set them apart from bacteria and archaea?
Well, the hallmark of eukaryotic cells is their internal complexity. Unlike bacteria and archaea, they have a true nucleus that encloses their DNA, and they have a whole system of internal membranes that create compartments and organelles for specific functions.
Right, organelles like mitochondria and chloroplasts are really what make eukaryotes so successful, right?
Absolutely. They are the key to their energy production and metabolic flexibility. And what's fascinating is that we now know these organelles have a very specific origin.
Endosymbiosis.
Yeah.
The idea that these organelles were once free-living bacteria.
Precisely. The evidence for endosymbiosis is overwhelming. Mitochondria, the powerhouses of eukaryotic cells, arose from a free-living, oxygen-respiring bacterium that was engulfed by an ancestral archaeal-like cell. This primary endosymbiotic event is considered a major turning point in the evolution of life.
It's amazing to think that a chance encounter billions of years ago led to such a profound transformation.
I know, right? And this symbiotic partnership was so successful that it became a defining feature of all eukaryotes. All extant eukaryotes either have mitochondria or have reduced forms of mitochondria like mitosomes or hydrogenosomes, or they carry genetic traces of an ancestral mitochondrion.
So basically, every eukaryotic cell we see today owes its existence to that ancient merger.
Exactly. And then in a separate lineage, we had another primary endosymbiotic event that gave rise to chloroplasts.
The photosynthetic engines of algae and plants.
Right. In this case, a eukaryotic cell that already possessed mitochondria engulfed a photosynthetic cyanobacterium. This led to the evolution of the Archaeplastida, the supergroup that includes red algae, green algae, and all land plants.
So we have two primary endosymbiotic events, each laying the foundation for a fundamental biological process. And then, to make things even more interesting, we have secondary endosymbiosis, right?
You got it. In secondary endosymbiosis, a eukaryotic cell engulfs another eukaryotic cell that already has a chloroplast.
So it's like endosymbiosis squared.
Exactly. This process has happened multiple times throughout evolutionary history and has led to a remarkable diversification of phototrophic eukaryotes.
It's incredible how many layers there are to the evolution of these organisms.
Totally. For example, euglenids and chlorarachniophytes acquired their chloroplasts through secondary endosymbiosis with green algae, and this is reflected in the structure of their chloroplasts, which have more than two membranes.
More membranes, more history.
You got it. And then we have groups like the alveolata, which includes dinoflagellates and apicomplexans and the straminopiles, a very diverse group, which both acquired their chloroplasts through secondary endosymbiosis with red algae.
So we have a good understanding of how mitochondria and chloroplasts came to be, but what about the origin of the eukaryotic cell itself? Is that still a bit of a mystery?
It is. While endosymbiotic theory explains the origin of these key organelles, the steps leading to the first eukaryotic cell with its nucleus and internal membrane systems remain an active area of research.
What's the current thinking on that?
The prevailing view is that the eukaryotic cell emerged from a symbiotic partnership between an archaeon and a bacterium.
A fusion of two very different types of cells.
Right. And this fusion likely involved a complex exchange of genetic material, leading to what we now call a genetic chimera, a mosaic of genes from different ancestors.
A fascinating puzzle.
Yeah.
But once those core eukaryotic features were in place, things really took off, right? There was a huge diversification of microbial eukaryotes.
Exactly. And modern phylogenetic analyses, using tools like whole genome sequencing, have really transformed our understanding of the relationships between these organisms. We now recognize 5 major supergroups of eukaryotes.
5 major branches on the eukaryotic tree of life.
Right. We have the Archaeplastida, which we already mentioned. Then there's the sarclade, which includes the stramenopiles, alveolata, and rosaria.
Okay.
And then we have the excavates, the amoebozoa, and the opisthocanta.
And the opisthocanta includes both fungi and animals, right? That always blows my mind. We're more closely related to fungi than plants.
It is pretty wild when you think about it. But the genetic evidence is clear, and it suggests that fungi and animals shared a common ancestor that diverged from the other eukaryotic lineages quite early on.
And our notes mention that this early diversification of eukaryotes might have been triggered by the acquisition of mitochondria.
That's a fascinating hypothesis. The idea is that the increased energy provided by mitochondria allowed early eukaryotes to experiment with new forms and lifestyles. It's like they suddenly had the power to evolve more complex structures and explore new ecological niches.
Makes sense. And speaking of energy, how do these microbial eukaryotes obtain the fuel they need to survive?
Well, most of them are either chemoorganicotrophic, meaning they get their energy from organic compounds, or they're phototrophic, meaning they use light energy for photosynthesis.
Okay.
And given that they all have mitochondria or some derivative thereof, most are also obligate aerobes. They need oxygen to survive.
All right, that gives us a good overview of what defines microbial eukaryotes and how they evolved. So now let's dive into our first major group, the protists.
Sounds good.
And our notes describe protists as an informal term. What does that mean?
It means that protist isn't a true taxonomic group in the sense that it doesn't reflect a single branch on the tree of life. Instead, it's more of a catch-all term for eukaryotic microorganisms that aren't plants, animals, or fungi.
So basically, if
Well, the excavates are a supergroup of eukaryotes that share some specific cytoskeletal features. And many of them have a characteristic excavated feeding groove, which is where they get their name.
makes sense. And our notes list 4 key groups within the excavates: diplomonads, parabasalids, kinetoplastids, and euglenids.
Right.
Let's start with the diplomonads. What are they like?
Diplomonads are interesting because they lack true mitochondria. Instead, they have these reduced organelles called mitosomes, which don't produce ATP.
So they're living without the main power source of most eukaryotes.
Exactly. They've adapted to survive in anoxic environments, getting their energy through fermentation. A good example is Giardia, an intestinal parasite that causes giardiasis.
Oh, I've heard of that. Not fun.
Definitely not. Next, we have the parabasalids, which also lack traditional mitochondria.
Okay.
But instead of mitosomes, they have hydrogenosomes, another type of reduced organelle.
And what do hydrogenosomes do?
They produce ATP through fermentation, just like mitosomes, but they release hydrogen gas as a byproduct. One of the best-known parabasalids is Trichomonas vaginalis, a sexually transmitted parasite. It actually has a surprisingly large genome for a single-celled organism.
And it also lacks introns, right? Those non-coding sequences within genes.
Yes, which is unusual for a eukaryote. It seems to have streamlined its genome over time.
A very efficient parasite. Okay, what about the kinetoplastids? What sets them apart?
Kinetoplastids are named after their kinetoplast, a dense mass of DNA found within their single large mitochondrion. This group includes some pretty nasty parasites, like Trypanosoma brucae, which causes African sleeping sickness,
Right.
and Trypanosoma cruzi, which causes Chagas disease.
So a small group, but with a big impact on human health.
For sure. And then we have the euglenids, perhaps the most well-known group within the excavates.
Euglena, that classic single-celled organism from biology class.
Exactly. And what's cool about euglenids is their metabolic flexibility. Some, like euglena, have chloroplasts and can photosynthesize, but they can also switch to a heterotrophic lifestyle, absorbing organic nutrients or even engulfing food particles.
So they can be both plant-like and animal-like, depending on the circumstances.
Pretty versatile, and that makes them very successful in a variety of freshwater habitats.
All right, next up we have the alveolata. What's the defining feature of this group?
Alveolata are named after their alveoli, which are small membrane-bound sacs that lie just beneath their cell membrane. These alveoli give them structural support and may also play a role in other cellular processes.
Okay. And within the alveolata, we have three major lineages: ciliates, dinoflagellates, and epicomplexans.
Right.
Let's start with the ciliates. I remember these guys from microscopy labs, covered in those tiny hair-like structures.
You're thinking of cilia, and those cilia are what makes ciliates so fascinating. They use them for both movement and feeding.
So they're like tiny rowboats zipping around and sweeping food into their mouths.
Pretty much, and one of the classic examples of a ciliate is paramecium.
Right, and ciliates also have two types of nuclei, don't they?
They do. They have micronuclei, which are involved in sexual reproduction, and macronuclei, which control the day-to-day functions of the cell.
So a division of labor within the nucleus itself.
Exactly. And while most ciliates are free-living, some are parasites, like Balantidium coli, which can infect humans and cause dysentery.
Not good. Okay, what about dinoflagellates? I always associate them with those bioluminescent waves and red tides.
You're spot on. Dinoflagellates are a fascinating group. They're mostly marine, and many of them are photosynthetic thanks to those chloroplasts they acquired through secondary endosymbiosis with a red alga.
Right.
They have two flagellas, which cause them to spin as they move through the water.
Like tiny tops.
Exactly. And while many dinoflagellates are free-living, some form symbiotic relationships with other organisms, like corals.
Right.
But dinoflagellates can also be problematic. Certain species produce toxins that can harm marine life and humans.
That's what causes those harmful algal blooms or red tides.
Exactly. Species like Goniolacus and Physteria can cause massive fish kills and contaminate shellfish.
So they can be both beautiful and dangerous.
For sure. And then we have the epicomplexans, which are all parasites.
And they're named after their apical complex.
You
Right?
got it. The apical complex is a specialized structure at one end of the cell that helps them penetrate host cells. Unlike the other alveolata, apicomplexans don't have cilia or flagella in their adult stage. They rely on other means to move from host to host.
Sneaky. And this group includes some major human pathogens, right?
It does. Plasmodium, the parasite that causes malaria, is perhaps the most notorious apicomplexan. But there's also Toxoplasma gondii, which causes toxoplasmosis, and various Eimeria species that cause coccidiosis in animals.
So a group with a huge impact on human and animal health.
Absolutely.
Okay, moving on to the stramatopiles. I know this is a very diverse group. What unites them?
The defining feature of stramatopiles is the presence of hairy flagella. At some point in their life cycle, they have flagella with these fine, hair-like projections.
Interesting. And this group includes oomycetes, diatoms, golden algae, and brown algae.
It does. And once again, we see both photosynthetic and non-photosynthetic lineages within a single group.
Convergent evolution strikes again.
Exactly. So let's start with the diatoms. They are incredibly important primary producers in aquatic ecosystems.
Right, the tiny algae with those intricate glass shells.
That's right. Those shells, called frustules, are made of silica, and they exhibit a stunning array of shapes and patterns.
Works of art at the microscopic level.
Totally. And those frustules are also incredibly durable, which is why diatoms have such a rich fossil record.
So they give us a glimpse into ancient oceans and the life that thrived within them.
Exactly. Now, what about oomycetes?
They're also called water molds, right? But our notes say they're not true fungi.
That's right. They were once classified with fungi because they have filamentous growth and absorb nutrients from their surroundings. But molecular data has shown that they're actually more closely related to other stramatopiles.
So another case of convergent evolution.
Exactly. And despite their name, not all oomycetes live in water. Some are important plant pathogens.
Like Phytophthora infestans, which caused the Irish potato famine.
Exactly. And there are also species of Pythium and Albugo that cause various plant diseases.
A reminder that even microscopic organisms can have huge historical and economic impacts.
For sure. Now let's move on to golden algae.
Golden algae, is that because of their color?
It is. Golden algae, or chrysophytes, get their color from the pigment fucoxanthin, which masks the green chlorophyll in their chloroplasts. They're mostly unicellular, and they're found in both freshwater and marine environments.
Okay.
While most golden algae are photosynthetic, some can switch to a heterotrophic lifestyle when conditions are unfavorable, and some even form colonies like Dinobrion.
So more metabolic flexibility.
Exactly. And finally, within the stramatopiles, we have the brown algae.
The big ones, the seaweeds.
You got it. Brown algae are almost entirely marine, and they include some of the largest and most complex protists, like kelp, which can form underwater forests.
Incredible. And they get their brown color from fucoxanthin, right?
They do. Just like the golden algae, it's a very effective pigment for capturing light energy in deeper waters.
Okay, we've covered a lot of ground with the stromatophiles. On to the Rosaria now.
Right.
What characterizes this group?
Rosaria are united by their thread-like pseudopodia. These extensions of their cytoplasm allow them to move and capture food.
Okay. And there are three main groups within the Rosaria: chlorarachnophytes, foraminiferans, and radiolarians.
Right.
So let's start with the chlorarachnophytes. They're another example of secondary endosymbiosis, aren't they?
They are, and they're particularly fascinating because they've retained a remnant of the green alga's nucleus within their chloroplast.
Wait, so there's a nucleus inside a chloroplast inside a eukaryotic cell.
Exactly. It's called a nucleomorph, and it's evidence of that secondary endosymbiotic event.
Layers upon layers of evolutionary history.
Yeah, I know, right? It's like a Russian nesting doll of cells.
Wow.
And the chloroplasts of chlorarachnophytes also have four membranes, another testament to their complex origin.
Wow. Okay, what about foraminiferans? I know they're important in paleontology.
You're thinking of their tests, or shells. Foramiferans, or forams for short, are marine protists that secrete these elaborate shells, often made of calcium carbonate.
And those shells fossilize really well, right?
They do, which makes them incredibly useful for studying past environments and climates.
These tiny time capsules.
Exactly. And many forams also host endosymbiotic algae, adding another layer of complexity to their biology.
It seems like symbiosis is a recurring theme in the microbial world.
It really is. And then we have the radiolarians, another group of marine protists with intricate skeletons.
But their skeletons are made of silica, right? Like the diatoms.
That's right. And they often exhibit beautiful radial symmetry, making them look like tiny snowflakes or stars.
Amazing. And those silica skeletons also fossilize well, don't they?
They do. Radiolarians, like forams and diatoms, have left behind a rich fossil record that helps us understand the evolution of marine ecosystems.
Okay, moving on to the haptophyte.
Sounds good.
Now, this group might be less familiar to some folks. What are they all about?
Haptophytes are unicellular photosynthetic protists that are primarily found in marine environments. They have two flagella and two chloroplasts, which they acquired through secondary endosymbiosis with a red alga.
Okay.
But their most distinctive feature is their haptonema, a unique appendage that looks like a flagellum but functions differently.
How so?
Unlike a flagellum, which is primarily for movement, a haptonema is used for attachment and feeding. It's like a long, sticky thread that they can use to grab onto surfaces or capture prey.
Interesting. What's a key group within the haptophytes?
The coccolithophores are a major group of haptophytes, and they're incredibly important for global climate.
How so?
Well, coccolithophores are covered in these tiny plates made of calcium carbonate called coccoliths, and when they die, those coccoliths sink to the ocean floor, forming massive deposits of chalk.
So they're playing a role in the carbon cycle, removing carbon dioxide from the atmosphere.
Exactly. They're also incredibly abundant, forming a major component of marine phytoplankton.
Small organisms with a big impact.
Absolutely. And their life cycle is quite interesting, too. They alternate between haploid and diploid phases, and the two phases look very different.
Making it hard to study them, I imagine.
It can be. Okay, we're on to our last group of protists, the amoebozoa.
Dish shapeshifters.
You got it. Amoebozoa are characterized by their lobe-shaped pseudopods, which they use for movement and feeding.
Right, those temporary extensions of their cytoplasm.
Exactly. And within the amoebozoa, we have three main groups: gymnamoebas, entamoebas, and slime molds.
Okay.
Gymnamoebas are the classic free-living amoebas that you might find in freshwater ponds or soil. Amoeba proteus is a good example. They feed by engulfing food particles with their pseudopods.
Pretty straightforward. What about entamoebas?
Entamoebas are parasites of animals, and Entamoeba histolytica, which causes amoebic dysentery, is a major human health concern in some parts of the world.
Another example of a tiny organism that can cause big problems.
For sure. And then we have the slime molds, which are a bit of an oddball group.
Mm.
Well, they were once classified with fungi because they produce fruiting bodies that release spores, but we now know that they're not closely related to fungi at all.
So another case of convergent evolution.
Exactly. There are two main types of slime molds: plasmodial slime molds and cellular slime molds.
Okay.
Plasmodial slime molds, like Physarum polycephalum, exist as a single multinucleate mass of cytoplasm called a plasmodium.
Wow. So one giant cell with many nuclei.
That's right, and they can move around and engulf food particles just like amoebas. Cellular slime molds, on the other hand, spend most of their lives as individual amoebae, But when food is scarce, they aggregate into a multicellular slug-like structure that migrates to a new location and then forms a fruiting body.
That's amazing. So they can switch between a solitary lifestyle and a cooperative one, depending on the circumstances.
Exactly. And the best studied cellular slime mold is Dictyostelium discoideum. It's a model organism for studying cell signaling development and social behavior.
Okay, we've made it through the fascinating world of protists. Let's move on to the fungi, a kingdom that's closer to us than we might think.
Closer to us than plants, that is.
Exactly.
Fungi are a monophyletic group, meaning they share a common ancestor, and that ancestor is more closely related to animals than to any other group of eukaryotes.
Wild. So what are the key features that define fungi?
Well, they're non-photosynthetic eukaryotes, meaning they can't produce their own food, and they have rigid cell walls made of coatine, which is the same material that makes up the exoskeletons of insects.
Strong stuff.
It is. And most fungi are non-motile, meaning they don't move around, although they do reproduce via spores, which can travel long distances.
Okay.
And fungi play essential roles in ecosystems as decomposers, breaking down dead organic matter and recycling nutrients. They're also important symbionts, forming mutually beneficial relationships with plants.
Like mycorrhizae, the fungal networks that help plants absorb nutrients from the soil.
Exactly. And some fungi are also pathogens, causing diseases in plants and animals.
So a diverse group with a wide range of ecological roles.
Very much so. And their structure is quite unique, too.
How so?
The basic building block of most fungi is the hypha, a long branching filament, and a mass of hyphae is called a mycelium.
So the mycelium is like the body of the fungus.
Exactly. And those hyphae can be either septate, meaning they're divided into compartments by cross walls, or coenocytic, meaning they're multinucleate and lack those cross walls.
Okay. And how do fungi reproduce?
They can reproduce both asexually and sexually. Asexual reproduction can occur through fragmentation of the mycelium, budding in yeasts, or the production of asexual spores called conidia.
Boy.
Sexual reproduction is a bit more complex and varies depending on the fungal group, but it generally involves the fusion of hyphae from two compatible individuals, leading to the formation of sexual spores.
Right.
And those sexual spores are often more resistant to harsh conditions than asexual spores, which helps them disperse to new environments.
Makes sense. And our notes mention that fungi have largely lost flagella, like animals.
That's right. Most fungi don't have flagella at any stage in their life cycle, except for the Chytridiomycota, which are a more primitive group of fungi that retain flagellated spores.
So another link to the animal kingdom.
Exactly. Okay, let's talk about some of the major groups of fungi. We'll start with the Microsporidia.
Okay.
Microsporidia are obligate intracellular parasites, meaning they can only survive inside the cells of their hosts. And they've undergone significant evolutionary reduction, losing many of the features that are typical of other eukaryotes.
Like their mitochondria.
Exactly. They lack mitochondria entirely, relying on their hosts for energy production.
So they've streamlined themselves for a parasitic lifestyle.
Exactly. And their genomes are also very small, containing only the essential genes for survival and reproduction.
Fascinating. Okay, what about the Chytridiomycota?
Chytrids, as they're often called, are mostly aquatic fungi, and they're the only group of fungi that retain flagellated spores, which they use to swim through water.
So they're holding on to a bit of their evolutionary past.
They are. And while many chytrids are harmless decomposers, some are pathogens.
Like Batrachochytrium dendropatitis, the chytrid fungus that's been decimating amphibian populations worldwide.
Sadly, yes. It's a devastating disease, and it highlights the interconnectedness of ecosystems and the impact that even microscopic organisms can have.
For sure. What's next on our fungal tour?
The Mucoromycota. This group includes many familiar molds, like Rhizopus stolonifer, the black bread mold.
Oh yeah, I've seen that one.
It's a common sight on bread and other starchy foods, and it's a good example of a saprophytic fungus, meaning it gets its nutrients from dead organic matter.
Okay. And Mucoromycota are characterized by their zygospores, right?
Yes. Zygospores are thick-walled sexual spores that form when two compatible hyphae fuse together. They're very resistant to harsh conditions, allowing the fungus to survive unfavorable environments.
A survival strategy that's clearly been successful.
Absolutely. Next up are the Glomeromycota, which are a very important group ecologically.
Why is that?
Well, Glomeromycota form mycorrhizal associations with the roots of most land plants.
Right, those mutually beneficial relationships where the fungus helps the plant absorb nutrients from the soil.
Exactly. And Gromromycota are particularly good at this. Their hyphae penetrate the plant's root cells, forming structures called arbuscules, which provide a large surface area for nutrient exchange.
So they're essential for the health and productivity of terrestrial ecosystems.
Absolutely. Without Glomeromycota, most plants would struggle to survive.
Wow. Okay, let's move on to the Ascomycota. I know this is the largest and most diverse group of fungi.
It is. The Ascomycota, or sac fungi, include yeasts, molds, morels, truffles, and many plant pathogens.
Wow.
They're characterized by the formation of ascospores, which are sexual spores that develop inside sac-like structures called asci.
Okay. And I know one of the most famous Ascomycetes is Saccharomyces cerevisiae, baker's yeast.
Right. Saccharomyces cerevisiae is a model organism for studying eukaryotic cell biology and genetics, and it's also incredibly important for baking, brewing, and winemaking.
A true multitasker. And it has a very interesting mating system, right?
It does. Saccharomyces cerevisiae has two mating types, designated as A and alpha.
Okay.
And a yeast cell can switch its mating type by replacing the active genetic information at a specific locus with silent copies of the genes for the opposite mating type. This ensures that there's always a compatible mating partner available.
A clever way to ensure genetic diversity.
Absolutely. And finally, we have the Basidiomycota, or club fungi.
The mushroom people.
That's the one. Basidiomycota include mushrooms, puffballs, shelf fungi, and plant pathogens like smuts and rusts.
Okay.
They're characterized by the formation of basidiospores, which are sexual spores that develop on club-shaped structures called basidia.
Right. And those basidia are usually located on the underside of the mushroom cap.
Exactly. And the mushrooms we see are just the fruiting bodies of the fungus. The main body is the mycelium, which grows underground or within the substrate that the fungus is feeding on.
So just the tip of the iceberg.
Exactly. Okay, we've covered a lot of ground with the fungi. Let's move on to our final group, the Archaeplastida.
The group that includes red algae, green algae, and land plants.
That's right. And all of these organisms trace their ancestry back to that primary endosymbiotic event where a eukaryotic cell engulfed a cyanobacterium.
Right. The origin of chloroplasts.
Exactly. So let's start with the red algae, or rhodophytes. They're mostly marine organisms, and they get their red color from phycobiliproteins, which are accessory pigments that help them capture light energy in deeper waters.
Okay.
And some red algae are economically important. They're the source of agar and carrageenan, which are used as thickeners and stabilizers in food and other products.
Right. And some red algae can also survive in extreme environments, like hot springs and acidic habitats.
They can. Species like Cyanidium caldarium and Galgeria sulphuraria are examples of extremophiles, organisms that thrive in conditions that would kill most other life forms.
Amazing. What about green algae? They're the closest relatives of land plants, right?
They are. Green algae and land plants share a number of key features, including the presence of chlorophylls A and B in their chloroplasts and the storage of starch as a food reserve.
Okay. And green algae come in a wide variety of forms, from single cells to large multicellular seaweeds.
They do. Chlamydomonas is a classic example of a unicellular green alga. Spirogyra is a filamentous green alga. Volvox forms spherical colonies. And ulva, or sea lettuce, is a multicellular green alga that's common in coastal areas.
A truly diverse group. And within the green algae, there's a specific lineage that's thought to have given rise to land plants.
That's right. The charophytes, or stoneworts, are a group of green algae that share a number of key features with land plants, including similar cell wall structures and reproductive processes.
So they represent a crucial evolutionary link. Remarkable. And I know there's some interest in using green algae for biofuel production.
There is. Certain species, like Botryococcus brownii, produce large amounts of hydrocarbons, which could potentially be used as a renewable source of energy.
So green algae could be a key player in a sustainable future.
Absolutely. And that brings us to the end of our deep dive into the world of microbial eukaryotes.
Wow. We've covered so much ground, from the origin of eukaryotic cells to the incredible diversity of protists, fungi, and algae.
We've explored the impact of endosymbiosis, the key features of each major group, and the ecological roles of these often overlooked organisms.
And we've seen how they've shaped the planet and continue to influence our lives in countless ways. We've covered all four main sections of our outline, hitting the major points, theories, findings, and examples within each. Organelles and phylogeny, protists, fungi, and Archaeplastida, all done.
Yep.
From the energy boost provided by mitochondria and the photosynthetic power of chloroplasts to the remarkable adaptations of excavates, alveolates, striminopiles, rosaria, haptophytes, and amoebozoa, to the fascinating diversity of fungal forms and lifestyles, and finally to the evolutionary journey from ancient algae to the plant kingdom we know today, we've delved deep into this vast and essential realm of life.
So as we wrap up, here's a final thought for you to ponder. If endosymbiosis played such a crucial role in the evolution of eukaryotes, what other major evolutionary transitions might have been driven by similar symbiotic partnerships?
Food for thought indeed.
Thanks for joining us on this deep dive. Until next time, keep exploring the